welcome to The Explainer. Today, we're diving into the brilliant and incredibly complicated world of CRISPR gene editing. We're right on the edge of being able to rewrite the code of life itself. But what if the very thing we do to make our gene edits perfect actually introduces a hidden danger, one we never even saw coming? You know, this right here, this gets to the fundamental challenge of all gene editing. See, we've built these incredible molecular tools, these scissors that can cut DNA with just breathtaking precision. But the real make-or-break moment, that happens after the cut. It's when our fancy tech steps back and the cell's own ancient, stubborn machinery takes over to patch things up. So let's talk about that promise. A technology like CRISPR-Cas9 is, well, it's nothing short of revolutionary. Its main job is to act like a programmable GPS for our genome. It finds a super specific sequence of DNA and makes a clean, precise break. That break is called a double-strand break, or a DSB. And for a cell, this is basically a 5-alarm fire. A broken chromosome, that's a catastrophic event. It can literally kill the cell. So right away, the cell's internal repair crews are scrambled. And this is where things get really interesting, because now the cell has to make a critical choice. And this slide really shows the core conflict we're up against. The cell has two completely different ways to fix that break. It's a fork in the road. One path is fast and messy, and the other is careful and precise. The big problem for us? The cell almost always wants to take the messy route. So on one side, you've got what's called Non-Homologous End Joining, or NHEJ. Just think of it as the cell's default setting. It's the emergency crew that rushes in and just glues the broken ends back together, fast. But that speed comes at a cost, often introducing little errors. On the other side, there's Homology-Directed Repair, or HDR. This is the one we want. This is the master surgeon. It's a super precise pathway that can use a template we give it to perfectly rewrite a gene and fix a mutation. And what's so crucial to get here is that the cell naturally defaults to that messy NHEJ pathway, mainly because it's always available. The precise HDR pathway we need for therapy, it's much, much rarer. So this sets up a constant molecular battle, a real tug-of-war inside the cell. You have proteins like 53BP1 pushing for that quick, messy fix, while other proteins like BRCA1, yes, that BRCA1, are trying to promote the careful, precise repair. So for years, the most logical strategies seem to be, well, to cheat. If the cell keeps choosing the wrong path, why not just block it off? The whole idea was to force the cell down the HDR pathway, the one we actually want it to take. And to do this, scientists developed something called small molecule inhibitors. Honestly, the best way to think of these is as tiny, custom-made roadblocks. They're chemicals designed to find one specific protein in that messy NHEJ pathway and just temporarily shut it down, kind of like putting the emergency crew on a forced coffee break. The strategy was just beautifully simple and really powerful. Step 1, use CRISPR to make the cut. Step 2, at the same time, add an inhibitor that blocks a key NHJ protein called DNA PK. And with its main repair route closed for business, the cell has no choice but to use that HDR pathway way more often. The result? A huge increase in our chances of getting that perfect therapeutic edit. And you know what? It worked brilliantly. Just look at this. Research in certain cells showed that using a DNA PK inhibitor, this one's called New7026, could increase the rate of that precise HDR editing by up to 4 times, a 400% improvement. I mean, this was a massive step forward. So problem solved, right? Well, as it so often goes in science, biology is rarely that simple. See, the thing about blocking a fundamental process in a cell is you might be turning off a lot more than you bargained for. Sometimes, solving one problem can create a much bigger, completely unforeseen one. This incredible success led some researchers to ask a really important, kind of nagging question. Okay, we know this protein, DNA PK, is a huge player in that NHEJ repair pathway. But what else is it doing in the cell? What other jobs does this first responder have? It turns out it's a gatekeeper against something deeply sinister called extrachromosomal DNA, or ecDNA. These are these rogue, circular pieces of our own DNA that have broken off the chromosomes. They can carry powerful cancer-causing genes, and we now know they're a major driver of aggressive cancers and treatment resistance. And here's the bombshell. A 2024 study in the journal Cancer Discovery found that inhibiting DNA PK, the very same protein we were blocking to make gene editing more precise, actually promotes the formation of these cancer-driving DNA circles. We were silencing the guardian of the genome. So this brings us right to the core dilemma, doesn't it? A single, totally logical action had two completely opposite effects. The intended outcome was fantastic, boosting the rate of perfect gene edits. But the unintended outcome? Potentially catastrophic. We were accidentally disabling a key defense mechanism against a known driver of cancer. Now, this startling discovery doesn't mean gene editing is a dead end. Not at all. Far from it. It just means we have to be smarter. It shows that we have to work with the cell's incredibly complex biology, not just try to brute force a solution against it. And this has sparked a whole new wave of innovation. Instead of blocking the bad pathway, the new goal is to actively boost the good one. This has led to really cool tools like HDR fusion proteins. Scientists are essentially bolting the helpful HDR repair machinery using proteins like CTIP or RAD52 directly onto the CRISPR system itself. It's like sending the master surgeon in with its own specialized toolkit, making it way more efficient without having to fire the entire first responder. And this push for smarter, safer editing is expanding all over the world. This isn't just happening in a few elite labs anymore. The story of DNA PK inhibitors, it's not a failure, it's a critical lesson. The path to safely and effectively editing our own DNA isn't about overpowering ourselves. It's about developing a profound respect for the intricate, billion-year-old systems that are already running inside them, and then learning to work in partnership with that complexity. You know, it leaves us with a final and pretty humbling thought. As we get closer and closer to mastering the technology to rewrite the code of life itself, how deeply do we first have to understand the ancient biological playbook we are attempting to revise?
Thanks so much for spending some of your time with us today. We build these videos for you, so your feedback is our compass. What's one subject you've been wanting to see a deep dive on? Let us know in the comments section. We read through your suggestions to decide what we film next. Don't forget to subscribe and turn on notifications so you don't miss our next upload. Stay curious, and we'll talk to you in the comments.